Good, thank you. So thank you very much for hosting me here, and congratulations for the initiative. It is really impressive. I'm speaking from London, and I moved to London to King's College 3 years ago with the idea of developing drugs for myocardial infarction and heart failure. So we are searching for drugs that protect the heart against DNA damage and then induce regeneration of the heart. And we based most of our research on high throughput screenings. And I'm saying this because this is relevant with what I'm going to say. And we were very well with our research. We have 2 new startups company, and suddenly COVID hit last year in March. And on the 8th of March, the laboratory closed, but at the same time here in London, all the university, the government and charities started to finance research on COVID and calling for scientists that could give a contribute to try to contribute. So we took the challenge and converted 5 people in my group on working on COVID. And what we were mostly intrigued since the beginning is try and understand why COVID-19 is such a unique disease in terms of clinical characteristics. For example, we don't have an explanation why other people are more affected. We don't know the substrate for hypoxia. We don't know why there is lung thrombosis. We don't know why there is loss of smell and taste. And so we decided to see if we could contribute by looking at pathology samples from these people who have a lot of experience in pathology for cardiovascular disease. And the University Hospital in Trieste, where I come from and where I was working before moving to London, has a very strong experience and history, I would say, over the last 8 years or so in performing post-mortem analysis. So 50% of the people who die there are analyzed post-mortem, and they continue to do work also during the early phases of the pandemic, which was quite rare. So we had the possibility of accessing samples of people who die of COVID. We have more than 300 samples at our disposal. Something that we found in the lungs of these patients was quite expected, so massive alveolar damage. Something was much less expected, that is a very high rate of thrombosis. In some people, you could see even thrombosis of the large pulmonary and artery veins. These are some of the first macroscopic images of this thrombosis. What was even more striking was that there was thrombosis in almost 90% of patients of the microvasculature. So these are a picture of the lungs. It's even difficult to believe that this was a lung for enormous fibrosis that occurs. But what was striking was the presence of basically thrombi in the small veins and arteries. And one characteristic of this thrombi was that they were heterosynchronous. So there were thrombi in advanced stage of organization, so fibrosis, together with thrombi that were much more fresh, which is an indication that there is a sort of endogenous stimulus for coagulation and thrombosis as compared to emboli coming from outside. There was a lot of virus in these people who died of COVID-19, hitting pneumocytes in the alveolar space. And in several instances, the virus, so this is in situ hybridization with SARS-CoV-2 specific probes, the virus was present in very abnormal, cytologically abnormal cells, for example, cells with more than one nucleus or with a very large cytoplasm. And this virus was expressing the spike protein. You see that some of these are very abnormal cells. But what hit our attention was the presence of a very large number of these cytologically abnormal cells, so cells with three nuclei or more, up to 20 different nuclei, as you can see in these pictures. You see these very abnormal cells, and these were mostly pneumocytes, as they were stained for napsin or surfactant A, which are two pneumocyte-specific markers. There are some other examples in these lungs or these cytologically monstrous cells with very, very many nuclei in this very, very large cytoplasm. This is a large area of fibrosis, lymphocyte infiltration, and the presence of these very big syncytial cells. You see them here or here in the right bottom corner. You see, sometimes these syncytia, include really many dozens of nuclei. So, based on this observation, we returned back to the laboratory here at the School of Cardiovascular Medicine Sciences in London, and obviously the first thing we thought that this was the product of the fusogenic capacity of SARS-CoV-2 spike protein. We certainly in this audience, you all know that the main difference between spike from COVID-2 compared to spike from SARS-CoV is the presence of these additional amino acid residues that give the capacity of the protein to be primed by furin proteases, which means that the protein that is present on the cell membrane, about 10% of the protein during virus infection is found in the cell membrane and escape the endoplasmic reticulum and the ergic. And this protein induces fusion. So the first thing we did was to clone and codon optimize spike protein from the Wuhan sequence and simply transfected this protein into cells. And what we found after 12 hours of incubation was, or culture, was the presence of these enormous syncytia. These big holes are even syncytia that are detached from the plate. I have to say that this is one of the first experiments we did already in the beginning of April last year, and when I saw it, I was even scared of thinking that such a powerful fusogenic protein is present in the lung of infected people. Just to show you the fusogenic capacity, this in green are cells expressing spike, observed by time-lapse microscopy for 5 hours, and you see that they roam around and they start fusing and forming these very large syncytia. These are some other images of these 5 hour time-lapse microscopy movies. You see formation of these very large syncytia. If you image these at a much lower magnification, so each of these blue dots is a nucleus of the cell. This is a culture of Vero cells. Vero cells express the ACE2 receptor. This is transfection of SARS-CoV-1 spike. You see basically this identical to control. This is SARS-CoV-2 spike. You see these big holes with the nuclei on one side or in the middle, and all these are syncytia. Even more powerful of forming syncytia is the MERS-CoV spike, where you see these huge syncytia. Unfortunately, we don't have pathology examinations of MERS lungs, as for religious reasons. There are no autopsies performed in these patients. This is how these syncytia look like. Nuclei are in blue, spike is in green, and the cytoskeleton of the cells is in red. So basically the cytoplasm of this syncytia is just a very thin layer of cytoplasm between two membranes, with the nuclei usually in the middle. So basically from this came the idea that perhaps COVID-19 is not simply a disease where there is a virus that enters the cells and kills the cells for replication, and probably is not also only a disease in which pathology is caused by hyperinflammation and hyperimmune response, but there could be a role for the presence of abnormal cells in the lung that drive pathology. So based on this idea, we, and having, as I said, the access to a high throughput screening facility, we started thinking whether we could find molecules that would block syncytia formation. And here in the facility, we have available to FDA EMA-approved drug libraries. They include approximately 3,800 libraries already approved for therapy for other indications. And so we set up a couple of assays for screening. The first assay is an heterologous syncytia assay. So basically, we took a cell type that doesn't express ACE2, so doesn't fuse, and we transfected it to express the spike protein. These are U2OS cells transfected with SARS-CoV-2 spike, and co-culture these cells together with the cell that instead expresses ACE2 Vero cells. And the first cell was loaded with the green quantum dots, and the second cell was loaded with the red quantum dots. So if you co-culture cells without spike, the two cell types stay together. You see cells with red quantum dots in the cytoplasm or green quantum dots. However, if a spike is around, then cells fuse, and then you see cells with many nuclei and the cytoplasm containing both red and green quantum dots. And this is a situation that's very amenable to quantification by high content microscopy. So you instruct the microscope to identify cells with more than one nucleus and the cytoplasm with the quantum dots of two colors. Another assay that we did was exactly identical to what I showed you before. So we take an expression vector for spike, we transfect cells, Vero cells, and we look for syncytia, and then we quantify these syncytia. This is how syncytia look like in the screening in the absence of drugs. There are 6 different wells at high magnifications. This is a beautiful syncytia, which spike is in green, and the cell contour is the cell shape is contrasting with cell mask. And these are the results of 2 screening we did with the 2 libraries we have available, the Press-to-Chemical Library and the MS Discovery Spectrum Collection Library. So every time here you see a dot, this is the effect of one specific drug, and if it is 0, it means no effect. And here the scale is a z-score, so these are standard deviations from the mean. So if you take all the drugs that are here, they are strongly inhibitory of the formation of syncytia, some with up to 5 standard deviations. This is how the top scorer of the screening looks like. So basically, these are the big syncytia in the control, and these are the cells that are still transfected to spike with spike, so they are green, but they don't fuse. And the top drug in one screening was Niclosamide, and in the second screening, the second drug in this screening was again Niclosamide. All these that are red are drugs that are effective at a very highly significant, so more than 2.58 standard deviation from the mean. And so we focused our attention on these drugs. And we selected 83 of these, basically we took all 83 of these. Out of these 83, we selected 43 because we wanted the drug that is also active in protecting cells against SARS-CoV-2 infection, not only that blocks syncytia. So in collaboration with Wendy Berkeley here at Imperial first and with Mike Melima at King's later, we tested first the drugs for protection from cell death. This is just an example of these experiments. So these are cells, very low magnification. So all these gray are gray wells are wells filled with cells, and these are the same after we give the virus. And these are the results of some of the drugs. Look at Niclosamide, for example. You see that even low concentration of Niclosamide protect the cells from death. So based on these criteria, then we did more refined virological assessments, and we found that the top 3 drugs that we selected, Niclosamide, clofazimine, and salinomycin, are highly protective against, inhibitory against the viral replication with an IC50 in the 100 nanomolar range for Niclosamide. Niclosamide is a drug that is used against tinea infection and tinea infestation of the gut, and I will speak about this later. Clofazimine is the top drug that is used against leprosy. Salinomycin is approved only for veterinary uses and antibiotic use in animals. Obviously, we were interested by understanding what are the mechanisms that relate to the formation of syncytia and the effect of these drugs. And here we were stuck until we ordered the drugs that were effective according to therapeutic classes. So these are the same results as before. However, the drugs are grouped by classes, and the 0 is here. The red line is highly statistical significance, or was on the left, the red line is very effective. And what we noticed without a surprise was that there were 5 histamine 1 receptor antagonists, 11 antipsychotics, 8 antidepressants, and then we didn't understand again what this means until we realized that all these classes of molecules that are active are molecules that block signaling through GPCR, G protein coupled receptor, coupled with G alpha Q proteins. And perhaps you remember that the G protein alpha Q version activates phospholipase C, which activates in turn IP3. And this is a powerful activator and opener of ligand-cutting channels for calcium. So basically, if you activate these type of GPCRs, you release calcium from the endoplasmic reticulum, and the drugs that block this effect then reduce the levels of calcium. So there was involvement of calcium somehow in syncytia formation. Again, we didn't understand why. And so we started the collaboration with an electrophysiologist here in London, Juan Burone, and he provided us a GCAM6 calcium sensor. Basically, this is a protein-based modeling that can be expressed in cells and becomes more fluorescent the more calcium is present around. And this is the effect what we see if we co-culture cells that don't fuse, so with the GCAM sensor, with the cell that express a spike. So basically in red, a cell that express spike, and in green, a cell that expresses the sensor. And you see that when the cells start fusing, there are these enormous oscillation of calcium around. This is an overnight view of this process. You see that star cells progressively fuse, and while they fuse, they activate enormous levels of calcium, like a Christmas tree, and that shows that really there is a massive increase in the levels of calciums. This thing can be measured, and so basically if you give spike, we increase release of calcium. If we give the drugs, we completely block this release. This is the 300 minutes observation. Each spike here, sorry for the double meaning of the word spike, is the increase of calcium oscillations due to the spike protein expressed in the cells. This is how the drugs work. So this is a DMSO control. So you see that the cell fuse and they have these calcium oscillations. If you give niclosamide, the cells are still there and they don't fuse and they don't have these calcium oscillations. And we know that calcium comes from the endoplasmic reticulum because we completely blunt this oscillation if you give tapsigargin or cyclopropiononic acid, which blunt release of calcium from the endoplasmic reticulum. So at this point, we know that basically there is a connection between the levels of calcium that is released from the endoplasmic reticulum, and obviously we couldn't understand how this has any relationship with syncytia formation. And reading the literature, we stumbled upon information about a very large screening performed 2 years ago by a group of German and US investigators who are searching for compounds that block bronchial cells contractions. That was a screening for drugs for asthma. And basically what they found, they were screening with more than 500,000 small molecules. And what they found were two molecules that were niclosamide and a close analog, that is nitazoxanide, that were very, very powerful at releasing contraction of smooth muscle cells from bronchi. And both drugs were found to be very selective and powerful inhibitor of a cell membrane protein called TMEM16A protein. So TMEM proteins are a family of 10 cell membrane channels that have the capacity of allowing chloride ion to get outside the cells, and some of the proteins have also the capacity to allow phosphatidylserine to go from the inner leaflet of the membrane to go to the outer leaflet of the membrane. We turned to our cells, the cells we were using in our syncytia assays, and tested the levels of all the TMEM16 proteins. What we found is that the protein that was expressed in all the cells that we were investigating was TMEM16F, which is the prototype of the scramblase type of these families. So the channels that allow phosphatidylserine to go outside the cells. And also we realized that all cellular events in biology that involve cell fusions are mediated by phosphatidylserine. Several of them are shown to be activated by TMEM16 proteins. For example, macrophages fusing into inflammatory giant cells do that through phosphatidylserine, or myoblasts fusing into myotubes, the same. Syncytia trophoblasts in the placenta use TMEM16F. And in fact, what we found is that if you take cells, we give ionomycin. Ionomycin activates calcium, and we have a lot of annexin 5 positivity, so for phosphatidylserine outside. If you give an siRNA TMEM16F, we completely block this kind of activity. If you give spike to cells, there is a very large amount of phosphatidylserine on the outer membrane of these syncytia. Here, syncytia are stained, are coated with a co-transfected M-cherry nuclear protein. And all syncytia in the lungs of patients are positive for annexin 5. So basically, the model now becomes that there is calcium induced by spike, and this calcium activates these channels on the membrane. These are calcium-activated chloride channels and scramblases. And upon this activation, there is phosphatidylserine exposure on the outer membrane of the cell. In fact, if you take spike, you transfect cells, you have big syncytia. If you knock down by an siRNA 16F, you block syncytia formation, more or less similar to what you have if you knock down the receptor.
Mauro, sorry, this is a 2-minute warning.
Yeah, thank you. I'm almost closing. If you give if you activate phosphatidylserine with ionomycin, give the drug, you block the effect. And basically, the idea is that all the drugs that we have selected are working because they target this mechanism, either indirectly as the anti-histamine 1 receptor antagonist, antipsychotics with block calcium, or like niclosamide, nitazoxanide, because they block the channels. I believe this is important for several reasons in explaining the pathogenesis. First, because there is chloride secretion, and chloride secretion, which is activated by spike. This is released, there are chloride currents, and so this is chloride released after you give spike. And this can explain the sudden edema that occurs in patients with SARS-CoV-2. Second, because the TMEM16F is the main protein that can amplifies platelet aggregation more than 10,000 times. And in fact, if you take a pseudotype particle with a spike and you expose platelets to this, you increase the capacity of platelets to aggregate compared to a standard VSVG pseudotype lentiviral vector. If you take cells expressing spike, you expose them to platelets, platelets aggregate on top of these cells. And third, because if you look at the olfactory epithelium, which is the inner part of our nose, there are, in the patients who die of COVID-19, but I suspect also in patients who don't die but lose smell and taste, there is a lot of viral infection. This is immunohistochemistry for the N protein, nucleocapsid protein for spike in these cells. So you see a lot of viral infection. And these are the same cells that use for olfactory transduction TMEM16A, TMEM16F. So this is a perfect mechanism also to explain loss of smell and taste. And obviously this reinforces the interest in Niclosamide as a drug to be repurposed for COVID-19. There are a few trials in the United States, a couple here in the UK, one we have inspired also in India recently with the Niclosamide for these patients, and we will see if it really works in patients with COVID-19 by reducing severity of the disease, perhaps also these accessory symptoms of the disease. As I said, this is sort of work mainly conducted by very fantastic postdoctoral scientists here at King's. During lockdown period, we work really for 4 months, day and night, and weekends. Here, all the pathology is by Rossana Bussani, the University of Trieste. This is a collaboration with the ICGB, in particular Serena Zacchini. I already mentioned the virologist who helped us, Wendy Berkeley and Mike Melim here in London. And I thank you very much for your attention. I'm happy to take questions if some time remains.
Thank you, Mauro. I just would like to remind the audience members to please raise your hands, and I will call on you and unmute you in the order that I see your hands. If I could start out with a couple of perhaps naive questions. In your original assay where you expressed spike protein, what fraction of the cells actually fuse with one another? And then the same question with respect to your cell fusion 2-color assay. How does that fusion affected by the expression level of the spike protein?
Yeah, these are not at all naive as questions. Fusion is extremely efficient. So we had to throw away experiments since we just left the protein to be expressed for 2-3 hours more than the usual 12 hours we express it. So it is a tremendously efficient process, and we can have really big lakes of cells even in overnight culture. And heterologously, that depends on the ratio by which you plate the cells. But if a cell expresses spike and finds a cell with a receptor, then fusion is almost inevitable. It is a dramatically efficient process.
And sort of along those same lines, does it matter? I assume it does. When you
sorry, what?
Does it matter when you give the drug to the cells? Does it have to be prior to spike expression?
Oh yes, yes. It has to be prior. It has to be prior.
Okay.
We try to see what happens if we give it after syncytia have started. They don't revert.
Okay.
Fusion is a process that by which you need only a membrane and the spike protein. What the drug do is blocking the increase of syncytia. They don't block fusion of single cells to other single cells. They block formation of big syncytia.
Got it. We have a question from Jahar Bhattacharya. Jahar, you should be able to unmute yourself and ask your question.
Yeah, hi. That's a very interesting presentation. Thank you so much. My question is the ACE2 receptor a GPCR?
No, it is not. It is a single pass transmembrane protein with a single pass, short cytoplasmic tail, and then all the rest of the receptor is outside the cell.
So how does it? What's the connection to the GPCR again? I just missed that.
No, the GPCR, there is no connection. Every time you block a GPCR with an alpha Q, a G alpha Q, you blunt calcium levels in the cell. And this is the reason why you decrease syncytia. So you need calcium for the syncytia because calcium activates the TMEM16 proteins. So if you blunt calcium or you omit calcium, you don't have syncytia anymore.
No, I understand that, but I'm trying to follow the signaling pathway from the ACE2 receptor to the calcium. So once ACE2 is ligated by spike, you now get.
No, sorry, no, it's not. I mean, ACE2 is a docking protein for spike, but the effect on calcium is triggered by spike itself, independent of ACE2. So what we are finding in reality is that there is an interaction, a direct interaction, a direct activation of TMEM16 proteins, and TMEM16 protein then sustain calcium for release of calcium from the endoplasmic reticulum.
So are you suggesting that spike has to be internalized, and then that internalized spike interacts with TM? Yeah, I'm wondering how that interaction. Yeah.
No, that's a crucial point. We don't know yet. There are two possibilities we are pursuing. One is that spike is expressed in the endoplasmic reticulum, and this, and then it drives release of calcium from the endoplasmic reticulum. It is calcium that activates TMEM16 proteins. Another possibility is that it is the inbound virus that, binding to the cell surface, activates TMEM16 proteins. So, for example, TMEM16A has been found associated with the GF receptor, so it's not unlikely that there is also an association with the ACE2 receptor, or we don't know that yet.
Thank you very much.
Thank you.
Thank you, Mauro.